who, Dan? With a very
Dick
famous
Lewontin.
guy.
Yeah.
Leontin. Yeah, I didn't dare to say it was this one. Yeah, it's there. Yeah, with Leontin. And then he worked on evolution of mutation and evolution and epistasis in particular. That's how I got to interact with him. But he does a lot of other things, and I think today is going to be on very different subject. But he still works on epistasis, mutation, resistance to antibiotics, and also on mutation scan. But today is going to be a very different thing. And he's right now in Montpellier, so you can actually meet him in real. And he's going to be there until late June, I think, right?
I'm going to be here until late May. With luck, we'll be in France through the summer. But geographic distances are very distorted these days, aren't they?
Yeah, that's right. But
okay,
yeah, well, we'll take advantage. Contact him directly or me to meet him. Bye.
Please do. Please do. Thank you very much, Guillaume. And thank you to the organizers, Emmanuel, Nacho, Luis, and others for giving me the opportunity to present this morning. As Guillaume said, I'm a professor at Brown University in the US, and I'm here on sabbatical for the spring semester. This trip was meant to happen in the fall. It was then postponed for the obvious reasons. But I want to thank Guillaume in particular for doing the legwork and getting the key document that allowed my wife and me to get long stay visas, which allowed us to breeze through customs in Paris at the end of December. Many of our friends in the US said, why the hell are you going to France in the middle of a global pandemic? We feel just the opposite. You have to be somewhere. Why not the south of France? And we've really been enjoying ourselves here. In the lower left, you can see the view out my apartment window right now. We've been visiting the nearby countryside. Thank you, Guillaume, for the loan of the bicycles, which we've put to good use almost every weekend. And I also want to thank Rémi Froissart, who invited me, as Guillaume mentioned, to Montpellier almost 15 years ago and unwittingly planted the seeds for this long visit now. I'm very excited to be able to share the results I'm going to share with you now, in part because I think they're really exciting, and in part because I'm very interested in the possibility of developing some new experimental collaborations. So if some of the things that I talk about today resonate at all with questions you think about in the laboratory, I'd be very, very happy to explore the possibility of developing some formal collaborations. So before we get into the science, I want to acknowledge my collaborators. Almost all the work I'm going to do was performed by Eugene Rains, my postdoc, who's extremely talented. He's both a good experimentalist and a good theoretician. And he's also on the job market. So if you hear of any academic opportunities that might be of interest to him, please let me know. Scott Wiley was a postdoc a few years ago who was here, as they say, on the ground floor and made some key contributions to getting the theory off the ground. Paul Snagowski and Christina Birch are colleagues at other schools that have at different intervals made important contributions to our understanding. And none of this would have been possible without the generous support of the US National Science Foundation, which has paid Eugene's salary for the past 4 years. Okay, so I have an outline. Here is my outline. There are 4 things I want to talk about. First, I want to review classical results from theoretical population genetics, which find that the effect of population size on natural selection is only to influence the efficiency of natural selection. But in fact, we are now aware of several cases of mutations that exhibit what we call sign inversion. These are mutations that break the rule. They are not only affected in different efficiencies as population size changes, but indeed are affected in different directions by natural selection as population size changes. I'll review several published examples, starting with the one that we found, and then develop a synthetic understanding of the mechanism that pulls together all these examples, and then comment on future directions. Okay, so what's the classical theory? A new mutation's fate is stochastic in theory. In a finite population, a beneficial mutation can appear and actually will almost always be lost by bad luck due to the fact that the number of ultimately due to the fact that the number of offspring of every individual in the population has to be an integer. And so that drives realizations away from the deterministic expectations. So this is an expression that tells us the probability of fixation for a neutral mutation. It's equal to the reciprocal of the population size, or more generally, it's equal to its frequency. But we will primarily be interested in the probability of fixation of new mutations. In other words, mutations whose frequency is equal to the reciprocal of the population size. I should say, by the way, that for me, n, the population size, is the number of chromosomes in the population. I generally think and work in haploid organisms, so that's why it's 1 over n instead of the 1 over 2n one often sees in textbooks. Here's what this probability of fixation looks like, and we're going to see quite a few plots that are of this general form. The x-axis is the population size, or the logarithm of the population size, and the y-axis is the probability of fixation. Here, for a neutral mutation, it's the reciprocal of the population size, which is a straight line on this doubly log transformed plot. This is a modest elaboration, also from classical theory, for the probability of fixation of a mutation that has the selection coefficient other than 0, again, assuming that it starts at a single copy. And superimposing the form of that equation onto the previous plot, you can see that beneficial mutations have a probability of fixation that's better than the neutral benchmark, and deleterious mutations have a probability of fixation that's worse than the neutral benchmark, as you would expect. But importantly, selection is overwhelmed by drift when the population size is less than roughly the reciprocal of the selection coefficients. You can see the blue and orange lines converge, the green and orange lines converge, and that reflects the fact that drift becomes a much stronger process as population size gets small. In other words, selection becomes inefficient in small populations. Now, we're going to, throughout the talk, focus not on the probability of fixation, but on the normalized probability of fixation, because the benchmark, neutral benchmark itself responds to population size. That is to say, as shown here, the probability of fixation of a neutral mutation is not independent of population size, and we want to control for that effect. So we're going to focus instead on the probability of fixation of a mutation normalized or divided by the probability of fixation of a neutral mutation in that same population size, because the probability of fixation of a neutral mutation is 1 over n, dividing by 1 over n is the same as multiplying by n. So you are going to see a lot of plots today where the y-axis is not p-fix, but n-p-fix. And that will allow us to represent the neutral benchmark as a horizontal line. So this is the exact same analysis as I showed you on the last slide, except now everything is rotated, so the neutral benchmark is everywhere 1, but the same effect of drift, or the same interaction between drift and selection, is evident. And at small populations, when population size is roughly less than the reciprocal of the selection coefficient, the mutation tends to becomes ever more neutral with respect to its long-term fate. Okay? Sign inversion is a class of mutations that we discovered which break this rule. So here are 2 examples. One is from work of Martin Novak's on the evolution of cooperation. And this tit-for-tat cooperator has a probability of fixation that exceeds the neutral benchmark in small populations, but is less than the neutral benchmark in large populations. And the mutator story, which is where we began and will begin now, has the opposite form. It's selected against in small populations before becoming beneficial. I should say that we were very excited by this, was the formal nature of this result when we wrote the mutator paper and gave it the name sign inversion. When we went back to the literature, we realized there were several other papers that had described the phenomenon, but hadn't really appreciated, I would say, hadn't really appreciated the evolutionary novelty of this non-monotonic response to population size. Okay, so let's now look at the several published examples of mutations that exhibit this phenomenon called sign inversion. To begin with, I'm going to tell you, I'm going to spend a little bit of time talking about the evolution of mutation rate, both because that's where we entered the field, but also because it's the simplest model to understand. And the first thing one has to appreciate is that mutation rate in an organism can evolve. Mutation rate is determined by the particular allelic constitution of an organism at the loci that are responsible for DNA replication and repair. So you can imagine a mutation in a DNA replication or repair locus could affect the mutation rate. And this was first made crystal clear in early work coming out of Rich Lenski's lab. So if you're not familiar with this experiment, Rich had the very clever idea over 30 years ago to start 12 replicate populations of E. coli from an isogenic ancestor. So at the beginning of this experiment, at generation 0, every cell in every one of these flasks was genetically identical, and they've been propagated faithfully every day by a 100-fold dilution. As of March of last year, they were at about 73,500 generations. They then put the whole experiment in the freezer on account of the pandemic. A great deal has been learned from this experiment, and I don't think Rich had a particularly clear focused question that motivated him at the outset, but it's really been a tremendous test platform for looking at fine-scale properties of evolution by natural selection. And for our purposes, this result from Paul Snagowski, only a few years after, I'm sorry, that's a typo, it should say 1997, but less than 10 years after the experiment began, 10,000 generations in, Paul discovered that 3 of the 12 lineages, 3 of these 12 replicate populations had evolved the mutation rate that's roughly 100-fold higher than the ancestors. So arrayed across the x-axis of these 2 plots are the 12 replicate lineages together with the ancestors. On the y-axis is the mutation rate that Paul measured. And what you can see is for 3 of the 12, there are 2 different mutation rate assays that Paul used, but for 3 of the 12 replicate lineages, the mutation rate is between 10 and 100 times higher than it had been. Why did this happen? This is the explanation that Paul developed, and it's certainly true. It's important to appreciate that Rich's experiment was built with cells that were incapable of genetic recombination. They were intentionally started with a genotype that was Rec A minus. It's not able to do genetic recombination. So all the evolution that's gone on in the system has been asexual. Now, this picture is meant to characterize the idea that a mutator can reach high frequency by genetic hitchhiking. The idea is that each horizontal line is a different genotype, and time proceeds from top to bottom. So at the top line, the oldest line in this representation, a single genotype by chance discovers a mutation that increases its mutation rate. That's shown by the red dot. Now, by virtue of the fact that it has a higher mutation rate, this individual organism will be producing organisms that are genetically more different from it than typical organisms' offsprings are, and there's some probability that this individual will produce an offspring that has a beneficial mutation, shown by the green square. Because these cells are asexual, that beneficial mutation will thus be genetically linked to the mutation responsible for the higher mutation rate. And if the green beneficial mutation succeeds in spreading through the population, modulo the stochastic phenomenology I described for you 10 minutes ago, then as it increases in frequency, it will carry with it this linked mutator allele, which thus can spread through the population. And we're now essentially 100% sure that that explains why the mutator spread through those 3 replicates of Rich's experiment, and in subsequent years, several other mutators have been discovered. So we became interested in the question of how the probability of the fixation of a mutator might respond to population size. And I'm going to tell you the story the way we wrote it up in the paper, but I will admit, which is to say, I'm going to first show you some algebra, and then I'm going to show you some computer simulations that validate the algebra, and I'm going to show you a big experiment. In fact, as is often the case, the truth is a little bit messier. Eugene was playing around with some cells and trying to do some pilot experiments for the evolution of mutation rate. And he'd been doing his work in flasks when he was a graduate student. He wanted to do them in 96-well microtiter plates so he could get a little bit of economy of scale. And the mutator, which reliably evolved in the flasks, wasn't evolving in the plates. And we thought, well, maybe it's something about the abiological differences in the environment. And then we started to think carefully, and this is really Scott's pivotal contribution, Scott Wiley's critical contribution, that maybe population size matters. Okay, The way the story is told today is that we assume that in asexuals, mutators can fix by one of two processes. They can hitchhike with a beneficial mutation that then fixes. That's the picture I just showed you. And here's a schematic of the math behind that. X-axis is the log of the population size. Y-axis is this NP fixed quantity, the neutral benchmark as before. Imagine that the beneficial mutation that the mutator creates has a fitness advantage of 10%, then except for the fact that it was produced by a mutator, its probability of fixation is shown by the blue line. Well, the difference is that the mutator need not produce a beneficial mutation. Most mutations are deleterious. So most mutations that mutators make are deleterious. So the probability of fixation as a function of population size conditioned on the appearance of a beneficial mutation has to be downweighted by the probability of that event. That's the conceptual idea that we had. And so here's the algebra a little bit more formally. The probability of fixation by hitchhiking is given by the classical quantity on the right, but normalized by the probability of a beneficial mutation. At the same time, mutators can fix by drift in spite of the fact that selection generally acts against them. Again, most mutations are deleterious, which means most mutators will be selected against. But that doesn't mean that they won't fix. Just as beneficial mutations have a probability of fixation less than 1, deleterious mutations have a probability of fixation greater than 0. And this is a result from the good old days, a classical result that tells us that the selection that takes advantage of the fact that the selection coefficient acting against the mutator as a consequence of this recurrent deleterious mutational load that it's producing is given by the difference in mutation rates. So this is the classical probability of fixation, and the selection coefficient it's a negative selection coefficient, and its magnitude is given by the difference between the mutation rate of the mutator and the mutation rate of the wild type. Interestingly, it's independent of the distribution of fitness effects among these deleterious mutations, and that's a result that goes back to Haldane. It's quite a lovely result. Okay, so here's what that looks like in graphical form. Again, x-axis is the population size, y-axis is NP fix. The probability of fixing by drift in very small populations is equal to the neutral benchmark. But as population size goes up, selection against this deleterious load that the mutator primarily is producing becomes more efficient, and the normalized probability of fixation collapses. In total, then, the mutator can fix with probability given by the sum of these two probabilities. Mutators fix by drift or by hitchhiking, at the end of the day, they've fixed. So the probability is simply the sum of these two probabilities, written this way. And here's what that looks like. So I've simply taken the green and the red curves and added them together to get the blue. And here then are some computer simulations to validate our thinking. The algebra assumes that once a mutation occurs, once a selected mutation occurs, no subsequent mutations occur, which means the algebra disregards clonal interference. The reason why the simulations fall below the analytic expectation at very large population sizes is that there are multiple competing beneficial mutations in the simulations, and that when there are multiple competing mutations, the classical prediction for the probability of fixation overestimates the truth. Okay. Finally, Eugene went to the laboratory and did a Herculean experiment using the yeast Saccharomyces cerevisiae. He had constructs that were genetically identical, except one had a 20-fold higher mutation rate than the other. And then he did hundreds of replicate populations exactly the way Lenski did, except in microtiter plates, and managed the population size by managing the depth of his daily serial dilutions. So here are the results when the dilutions were shallow, which means the population size was large. He did almost 250 generations. He was able to track the frequency of the mutator by virtue of having also labeled these cells with fluorescent markers, and so he could count the cells efficiently on a fax machine. What you see, each blue line is the individual replicate, and the red dots are the averages across replicates. You can see the frequency of the mutator drops early because it's mostly producing deleterious mutations, but then it succeeds in reaching fixation in the vast majority of replicates, and across replicates, the average frequency of the mutator goes from 50% to 80%. As I told you at the outset, the probability of fixation for a neutral mutation in general is its starting frequency. So if the mutator were neutral, we would expect its ending frequency to be 50%. This 80% or so endpoint is a huge difference and very highly significant because of all the replicates that Eugene did. Here's the same experiment when the daily dilutions are very deep, which means the effective population size was small. The mutator goes down in frequency and never recovers. Okay, so sign inversion and mutators. Once we had the idea that this might be a phenomenon for mutators, long before we actually finished the paper, we began to look into the literature and discovered this example from Christina Birch's group of a couple years earlier. They were studying the evolution of a modifier that allows sexual recombination. These are really important and complicated questions, the evolution of sex, which are very difficult to experimentally manage. And so Christina is using a population of gene regulatory networks, which is a simulation platform that was first brought to evolutionary biologists by Andreas Wagner, and others, including Mark Siegel, have made a lot of progress here. Christina here is doing the same experiment that we were imagining. There's a homogeneous population of asexual gene regulatory networks. She injects a single sexual gene regulatory network. She does that thousands or hundreds of thousands or millions of times and counts the fraction of replicates in which the sexual gene regulatory network lineage takes over the population, and finds this same non-monotonic response to population size, sign inversion. Why is this happening? To understand, we have to go under the hood of what happens in sexual and asexual populations. So the first thing to appreciate is that sexual organisms suffer a fitness burden as a consequence of recombination. Recombination will disrupt favorable pairs of alleles or favorable groups of alleles across the genome. That's called the recombinational load. And what Christina's student, Alex Whitlock, did here was to follow the recombinational load in homogeneous sexual and homogeneous asexual populations before there's any competition, just to see what happens. Now, asexual organisms don't suffer any recombinational load, so those black dots are at zero across the entire time scale of the experiment. But the asexuals, you'll see, have a transient increase in recombinational load, but then evolve recombinational robustness, and in other words, the recombinational load goes down. Recombinational load is defined as the difference in fitness between parents and offspring mediated by recombination. Very interestingly, and I don't know whether this had been described before, Christina's group observed a correlated response in the deleterious mutation rate. In other words, the evolution of mutational robustness gives a correlated evolution of mutational robustness. Let me make sure I said that right. The evolution of recombinational robustness gives a correlated response in mutational robustness. So you can see that in the second, in the middle row here, in the top, that's now the top row. Here, both the sexual and asexual lineages evolve lower deleterious mutation rates. They evolve to places on the fitness landscape where mutations hurt less. But because the sexual organisms are also being driven to parts of the landscape where recombination hurts less, it turns out that there's an extra reduction in the deleterious mutation rate. And this in turn gives rise to an extra increase in equilibrium mean fitness. It turns out, essentially using that same result of Haldane's, that the fitness of an organism can be described simply as a function of its deleterious mutation rate. And because the deleterious mutation rate in sexual lineages is lower, the fitness is higher. So, sexual lineages that are invading an asexual population will transiently suffer a fitness deficit due to recombinational load, but if they survive, they will evolve increased mutational robustness and thus higher mean fitness, and can displace the asexual resident. And that's what's shown here. So this is an average over a bunch of populations, a bunch of replicates. The red line is the fitness of the sexual invader. It begins by dropping below the resident mean fitness shown by the horizontal dashed line, before eventually increasing because of this additional reduction in deleterious mutation rate, resulting in an increase in mean fitness. Third example is this tit-for-tat cooperator that you've already seen. Tit-for-tat is a game theoretic strategy that represents cooperation. And individuals that adopt the tit-for-tat strategy have high fitness if they interact with other tit-for-tat individuals. but low fitness if they interact with cheaters called always defect. And so Martin Novak described this effect. And here's how it works. So this mutation, this tit-for-tat mutation, experiences positive frequency dependent selection. It's deleterious at low frequency because it's mostly interacting with with cheaters, with always defects, and it's being taken advantage of. But as its frequency goes up, assuming the population is well mixed, it will begin to interact often enough with other cooperators that they together will enjoy the advantage of being cooperators and thus become beneficial. And so for the tit-for-tat cooperator to invade, it has to negotiate this gap between 0 and the critical frequency where it becomes beneficial, which we call F. It must rise to F, even though it's deleterious at those frequencies. It must rise to F by drift, while rare. And then if it's successful in that, it will almost surely fix because it'll become beneficial. So one way of thinking about, let's see if I can find my pointer. Is that the pointer? One way of thinking about this, where the tit-for-tat cooperator has a higher than neutral probability of fixation, is that we're only asking the tit-for-tat cooperator to drift from 0 to F, whereas the benchmark is being asked to drift from 0 to 1. Now, there's selection acting against the tit-for-tat cooperator in that interval between 0 and F. But if population size is small, that reduction in probability of fixation, because the total transit distance is less, can be more than offset. That's what's going on here. Okay. We now turn to the synthetic part of the talk. We were fascinated by these three models. We thought they seem in many respects different from each other. They also tickled our fancy because they are each models of evolution that are outside of the kind of common domain where we've been working. You know, as Guillaume mentioned, I've worked on epistasis, I work in drug resistance. These mutations that I think about have an unconditionally beneficial or deleterious effect, whereas what I've just described, each of these mutations has a rather bizarre pattern of influence on fitness. And what we realized, this is recent work of Eugene, so I'll give him credit. What Eugene realized is that each of these mutations have the property that they're beneficial in some carriers and deleterious in others. So let's review. Mutators are deleterious because they can be genetically linked to deleterious mutations, but they're beneficial in those individuals in which they're linked to beneficial mutations. The recombiner is deleterious because it suffers a deleterious recombinational load, but if it can survive long enough, it becomes beneficial because it then imparts a reduction in the deleterious mutation rate. And the tit-for-tat cooperator is costly when it's rare because it's competing primarily with cheaters, but becomes beneficial if the frequency becomes high enough that it interacts primarily with other cooperators. When we thought about this a little bit more carefully, we realized immediately that we could partition these models into two classes. The mutator has the property that a particular lineage of mutators is either beneficial or deleterious. This is a little bit of an approximation, but the mutator is introduced in the population, and we imagine it almost immediately produces a fitness-affecting mutation, either beneficial or deleterious. And we also, for the moment, assume that there are no further fitness-affecting mutations. So what that means is that this lineage, this particular lineage of mutators, is either beneficial or deleterious. And so in this picture here, you can see as time moves from left to right, the three arrows at the top indicate independent appearances of this between lineage fitness variable mutation, e.g. mutator. And two of the lineages are deleterious, and one of them is beneficial. And the deleterious lineages, in the deleterious lineages, the mutation remains deleterious for the life of the lineage. And in the beneficial lineage, the effect of the mutation is to be beneficial for the life of the lineage. In contrast, both the recombiner and the tit-for-tat mutations have the property that their effects change over time, and the same lineage, individuals in the same lineage will sometimes experience a fitness cost and other times experience a fitness advantage. In the case of the recombiner, the change is mediated explicitly by time. If they can hang on long enough that the deleterious mutation rate drops below that of the asexual resident. In the case of the tit-for-tat, it drops over time, but as a function of changing frequency. So we subdivided these three cases into three simple models. One that exhibits between lineage sign variability, one that exhibits within lineage sign variability mediated by time, and one that exhibits within lineage sign variability mediated by frequency, and simulated simplified abstracted versions of each of these models to learn more. So model one is this between lineage variance model. Every time this mutation is introduced with probability p-ben, the mutant lineage, the mutant and all of its offspring enjoy a fitness advantage s-ben, and with probability 1 minus p-ben, they suffer a fitness cost s-cost. Here are simulation results from that model. Here is the probability of drifting to fixation, that is to say, the probability of drifting even though in the case of 1 minus p-ben of individuals that suffer the cost. Here is the probability classically of fixation of a mutation with benefit s-ben, which we again have to downweight by the probability p-ben. So this should be very familiar to you because it really reads almost exactly the same as did the mutator story I told you before, except that we've replaced the probability of a beneficial mutation in the context of this mutator model with this abstracted idea of something good happening to this lineage. And as a consequence of p-ben, selection for this allele remains overwhelmed by drift at population sizes much larger than you would have expected. So let's see if I can find my cursor again. Maybe here we go. So here is where selection would be overwhelmed by, where as population size gets small, selection becomes overwhelmed by drift here. But in this case, selection doesn't start to influence, positive selection doesn't start to influence the fate of this allele until way out here. The time-dependent fitness model is very similar. All mutants suffer a fitness cost s-cost for the first t generations, after which they all enjoy a fitness benefit s-ben. Here are the simulation results, and here is the fitness function. As time goes along, we decided for the first 50 generations, this allele would have a 10% fitness cost, after which it would have a 10% fitness advantage. Here is the probability of fixation normalized by the neutral benchmark over, I think it's 10 million replicates per population size. Here is the probability of fixing in the first 50 generations, i.e. while this thing is still deleterious. There's probably an analytic result that we could have used for this purpose. It's probably buried somewhere in Kimura's work, but I couldn't find it. So we just picked these numbers out of our simulations. Here again is the probability of fixation if it weren't for this 50 generation lag before becoming beneficial. Here is the key quantity. This is different than in the between lineage model. These alleles, these lineages have to survive for 50 generations in the face of purifying selection. Again, there's probably an analytic result for this, but I don't know it. So we picked these values out of the simulations. Population size on the x-axis, the probability of surviving, one or more lineages surviving to 50 generations plotted on the y-axis. And now we just multiply the classical probability of fixation by these probabilities of survival to recover exactly the NP fix for the time-dependent model. And again, as a consequence of p survive being very small, you can see it's on the order of 10 to the negative second to negative third for these parameter values. Selection remains in favor of this allele, remains overwhelmed at population sizes much larger than you would have expected classically. And finally, the frequency dependent fitness case, all mutants suffer a fitness cost s-cost while below frequency f, after which they all enjoy a fitness benefit s-ben. Here are the results. And in the inset, you can see the fitness function. And now the x-axis is not time, but frequency. This model is a little bit different because unlike the past two models where the mutant can fix by drift even though it's costly, in this model, the mutant has to transit across the entirety of the frequency spectrum. So every lineage that succeeds has to survive to a high enough frequency to enjoy the benefit. So the algebra turns out to be a little bit different. But here's the normalized probability of getting from frequency 1 over n to f. And you can see it drops with n, and that's because selection is acting against the allele over that frequency range. Here's the probability of getting from F to 1 normalized by the appropriate neutral benchmark. I'm not going to take time to explain why the leading coefficients here aren't n. It's got to do with the expectations for a neutral mutation getting from 1/n to some arbitrary frequency f, and the probability of a neutral mutation getting from arbitrary frequency f up to 1. Take my word for it, this math is right. And what you can see is the probability of fixation conditioned on getting to the frequency is higher than 1, but the probability of getting to the frequency is crashing as n goes up. Here we've shown the probability unnormalized. This is simply the probability of getting from 1/n to f as a function of population size, and you can see it crashes with n as you would expect intuitively. Also, as you probably would expect intuitively, it's falling much faster than n. That's important. When we multiply the probability of reaching fixation, from 1/f at each population size by the probability of reaching f, we again recover the simulation results. So we feel very good. We feel that we understand the parts that are in common in all three models and the parts that differ between all three models. Let me review what they are. Again, as a consequence of this new term, the probability of getting, from, in this case, getting from 1/n to f, selection for the allele becomes, now it's a little bit different. Selection for the allele becomes overwhelmed eventually. This quantity is dropping so fast that even though this probability is continuing to go up, albeit slowly, the product of the two eventually drops below 1. So in each case, the probability of fixation relative to classical expectations is reduced by the probability of being beneficial. Here's a cartoon of what I've told you repeatedly. Again, x-axis is population size, y-axis is the normalized probability of fixation, the neutral benchmark is the horizontal dashed line, the red curve is the classical result if the effect is unconditionally beneficial. In the between lineage case, the probability that it is beneficial is independent of n, and so we simply multiply the classical result by some constant to get this example of sign inversion. And because the probability of being beneficial is independent of n, the net effect of this multiplication is that the NP fix for the mutation of interest has this concave up shape. In the case of the positive time-dependent model, the probability of survival quickly becomes roughly independent of n. So we again can roughly multiply by a constant and again recover a concave up response to population size in this quantity NP fixed. The positive frequency dependent case is a little bit different because the probability of the effect being beneficial collapses so quickly with n. When we multiply that probability by the classical result, if the effect simply were beneficial, we get this concave down response. Okay, that's all I wanted to say in terms of new work. The last little bit is simply speculation and future directions. The first point I want to make is that Eugene's insight that all cases of sign inversion are for mutations that exhibit this sign variable fitness effect. So sign variable fitness effects, that a mutation is beneficial for some carriers and deleterious for others, is necessary for sign inversion. It's not sufficient. You can cook the parameter values in such a way that even though the mutation is sometimes beneficial and sometimes deleterious, you still don't see sign inversion. But this idea that a mutation could have different effects, different fitness effects in different carriers, is one that was already in the air in my group. My former grad student, Chris Graves, wrote this nice paper in the annual review a few years ago, in which he explored the consequences for natural selection in general of variability in fitness effects. So he's not focusing only on mutations that are sometimes beneficial and other times deleterious, but rather more generally, mutations whose fitness effect varies. And his point was that essentially, as ours is in this work, that the classical expectations for the probability of fixation go out the window. And Chris really felt like the punchline was that the idea that we were selecting for the fittest goes out the window. He's not the first one to say that, and these ideas seem to be sort of in the vicinity of all of the studies of all these kinds of mutations that don't simply have an effect of increasing resistance against ampicillin by 2 log orders. In other words, mutations that don't have an unconditionally beneficial or deleterious effect. So we put this table in the paper to sort of point the way to sources of variability across lineages. So there's biology in the specific examples, spatial variation, temporal variation in environment, kin selection, multi-level selection, and these genetic factors. And Chris's point was we can partition these various sources of lineage variable fitness effects into those that are driven by environmental change, those that are driven by social change, and those that are driven by genetic change. And then we put a few examples of mutations, bet-hedgers, cooperators, et cetera. I just want to point out that I've just shown you sign inversion operates on 3 of these kinds of mutations. More generally, I think there's a whole domain of mutations whose effects on their carriers is not constant that are likely going to be involved in these very interesting non-classical population genetic effects. Indeed, this table motivated my group to proceed a little bit further down the road, and these are results of Eugene's from simulation results for the evolution of ploidy. So as always, population size on the x-axis, normalized probability of fixation on the y-axis. This is a diploid trying to invade, and you can see that it's favored at small populations, but depending on the dominance coefficient becomes deleterious at larger population sizes. And this, the last thing I'm going to share with you is work from a younger grad student in my group working on the evolution of bet-hedging. So bet-hedging, you saw a couple of slides ago on the slide from Chris's, in the table from Chris's paper. Bet-hedging is an evolutionary strategy to deal with varying environments. If the environment is invariant, then there's going to likely be one strategy that's optimal. But if if an organism doesn't know what environment it's going to find itself in, or what environment its offspring are going to find themselves in, it sometimes pays to hedge your bet and produce offspring that draw their behavior from a distribution of possibilities, in the hopes that one of those offspring will be really well attuned to the environment that happens to appear. Classically, this line of reasoning has led to the idea that natural selection will favor strategies that maximize the geometric mean fitness across strategies. It's a geometric mean because replication is multiplicative, not additive. So we multiply the succession of fitnesses that a lineage enjoys over time, and the average is weighted by the probabilities that each particular environment is encountered. So classically, we believe that if the geometric mean fitness of a bet-hedger is higher than the geometric mean fitness of the resident specialist, the bet-hedger will invade. What Maya has shown is that's true, but in a stochastic framework, even bet-hedgers whose geometric mean fitness is less than the wild type's geometric mean fitness, the bet-hedger can invade, but it exhibits sign inversion. So only in sufficiently large populations will these bet-hedgers with lower geometric mean fitness invade. And the geometric mean fitness of the bet-hedger can't be arbitrarily low, but there's a zone outside of the zone in parameter space previously recognized. And Luis, this is what I was thinking about during your talk in January. It seems to have to do with interactions between stochasticity and the environment, which is largely averaged away in the GMF treatment. But the stochastic succession of environments that a lineage experiences, coupled with the stochastic successes and failures of reproduction, seem to be what's driving this effect, but we really don't understand it yet. Okay, that's all I have to say. I think the time is good. I've told you about classical expectations for probability of fixation. I've introduced you to the idea of sign inversion that breaks that classical expectation. I walked you through 3 examples and kind of opened up the hood to see how they all worked. And then I offered you 3 much abstracted and simplified models that seem to capture the spirit of these 3 published cases, which allowed us to understand that the essential features of sign inversion are sign variability and the particular sensitivity to the probability that the mutation will be beneficial in its carrier. And then I talked about some future directions in my group. And as I said at the outset, I'm very interested in the possibility of future directions of working with some of you, if any of you have experimental systems or theoretical ideas that maybe articulate with some of this. So thank you for your attention, and I'd be happy to take questions.
All right, thanks, Dan. So we already do have a few questions. So people can keep asking their question. That's the first thing I will say in the tab. Then they can also vote for questions, which helps us choosing. But for now, I'll just think we just start by the person coming from the farthest. So, oh, okay. So it will be Ariane De Visser who had a question. So can Emmanuel, can you turn his mic on, please? Because I don't remember how to do that. So
there, you should be able to activate your microphone.
You hear me?
Yes.
Oh, good. Okay.
Hi, Aaron.
Hi, Dan. Good to see you.
Yeah.
Thanks for a very interesting talk. I was just wondering whether, so the population size dependence of in the mutator case,
Yes.
where mutators get selected against in small and selected for in large populations, that must be sensitive to the relative rates of deleterious and beneficial mutations, I guess.
Absolutely.
And does it also, but how much does it depend on the relative rate and how much on the absolute rates of both classes? I was trying to understand that.
Yeah, well, it's very sensitive to the relative rates. The probability of the probability that a mutator produces a beneficial mutation is the beneficial mutation rate divided by the total mutation rate. Mm-hmm. And we made the simplifying assumption that the beneficial mutation rate is vastly less than the deleterious mutation rate, which allowed us to approximate that as the ratio of beneficial to deleterious mutations. And in our simulations, we set that value at 1%. Eugene is now engaged in some experiments to try to measure that from those 300 populations at large and small population size. The rate at which the frequency of the mutator declines at the outset speaks directly to that question. And so Eugene's working on some follow-up experiments to answer that question. The sensitivity of the, so are you okay with that answer?
Yeah, I guess that's a clear answer for me. Yeah, thanks.
What's interesting to me is that the strength of the mutator, how much higher, so we assume that the ratio of beneficial to deleterious mutations is unaffected by the mutator itself. Mm-hmm. But the strength of the mutator, is it a 20-fold or 100-fold or 1,000-fold mutator? That doesn't enter into our algebra at all. And I don't fully, we explored that, which is of course to say Eugene explored that in a supplemental to the last paper in this area. But I don't have a deep intuitive understanding for how that quantity enters into any of what I've just told you.
Mm-hmm. Okay. Well, thanks for now. Yeah.
Okay. So for the next one, I think we can have a sort of debate or something like that. So prepare for the challenge of having Thomas Lenormand and François Rousset, who are both commenting on some aspects. So I guess the both of them can sort of explain what they meant.
Hi, I'm Thomas. So perhaps I can let François ask this question. He really worked on this a lot, and he is probably the best person to ask about it. But the main point was that it looks like it's very familiar, this issue of population size to models where you got positive frequency dependent selection, like in many social evolution models. So that was the point. But I'm sure François can make the point in more detail if you want. Perhaps I can instead ask another more open question. I was curious about the diploidy models at the end.
Yes.
I was wondering which case was it exactly? Was it just a time dependence type of situation whereby diploid accumulates some load? I don't know.
No, it's actually almost exactly the same as the mutator case. Diploids, if the mutation, so this is a model where all mutations are deleterious, and so full dominance reduces the mutation rate of the diploid relative to the haploid.
Right.
So it's exactly analogous to the first model that I talked about.
All right, thanks.
All right, so then there was this comment about François. Do you want to
Yes, please.
elaborate on this?
Yes, so first there was a comment that yes, this is known from diffusion models, this sign inversion for diffusion model with frequency dependence. But a more general question is, there is no question that this process can occur, but then what is the kind of synthetic result that we can obtain, considering that there are mutants with different penetrants and strengths of effects and so on?
So I'm ignorant of the social evolution models that you're describing. So my request is, could you please send me a short reading list or a long reading list to get me started in that literature? It sounds absolutely fascinating. I'm sure it will be very interesting to me and my students. And unfortunately, I didn't quite understand the question about underdominance. Could you, or overdominance, or dominance in general? Could you ask that again for me, please?
Oh, yes. So if heterozygotes are deleterious, but
yes.
So homozygotes are favored.
Yes.
When the mutant is rare, it is selected against, and when it is common, it is favored. So it's exactly what you are describing, and so
yes.
So there are diffusion formula for probability of fixation of mutants in that case, and the tit for tat case is exactly analogous to that.
Yeah, yeah, and so I mean the figures I showed for tit for tat, I just lifted the algebra from Martin's paper of 2004, and I don't mean to pretend that this wasn't known in his literature or in the literature that you're more familiar with than I, but only to make the point that, so far as I could tell, the kind of evolutionary implications, and in particular, the sort of theoretical idea that this is such a radical deviation from the classical expectations, so far as I know, is our main contribution. But yes, right. I think the underdominance case is fascinating. Thank you for that suggestion. I'd love to read some of that literature.
So then we have a question by Anne-Florence. So that's why you don't have to completely trust the question and answer tab, because there are people under fake IDs.
Yeah, thank you, Luis. Right, I mean, I think it's just because Guillaume sent me the link and somehow now I'm labeled as Guillaume. But yeah, thanks a lot, Dan, for the great talk. I was wondering, you showed us very striking cases where NP-fix can be larger or smaller than 1, depending on the value of n. I was wondering if you encountered many more situations where NP-fix is simply non-monotonic, but still remains below or above 1.
Oh. No. My reaction is, but if I just continue to increase n, if let's consider a deleterious mutation, starting from small n, it drops, NP-fix drops below 1.
Yeah.
Now it's non-monotonic, so it's increasing, but hasn't yet gotten up to 1.
Mm-hmm.
I would ask you, why don't we just continue to increase the population size? Won't it eventually cross 1? I
see, okay. So no cases where you have, for instance, a maximum and then it decays towards 1, for instance?
Maybe asymptotically. So the answer is no. I have not.
Okay.
I've not observed anything of that sort. And my intuition is it's bad enough to have it's enough of a complexity to have a change of sign of the first derivative. To have it happen twice
I see. Yes.
is just a very abstractly. That seems that's a lot of biology.
Okay, thanks a lot.
Thank you. Yeah.
Luis, we didn't hear you.
Okay, Florian, you can ask your question, I think, now.
Hi, Dan. Thank you for your interesting talk. I'm not sure that I've understood at a point, but maybe you explained that about the evolutionary outcomes that you've shown us. How does it depend on the balance between the deleterious and advantageous effects? So you have at a point this example where the deleterious effect is log 0.9 at first, and then it's 1.1. So how does it depend on this balance in your models?
Only qualitatively. So you're absolutely right. And it depends, but only qualitatively. And I'm going to just make an assertion, but I'm rather confident that so that was for the time-dependent selection model. And if it starts deleterious and then becomes beneficial, I can't see how there couldn't be sign inversion. But it's a matter of making the simulations run in a realistic, practical amount of time. Incidentally, and similarly for the frequency-dependent model, you may have noticed there's an asymmetry. Selection against the cooperator was only 1% below the critical frequency and then jumped to 10%. And the reason is very simple. If the selection against, and we also made the critical frequency rather small, if selection against the cooperator is stronger, it gets killed in so many replicates that it just becomes a pain to finish the simulations. I will say that non-mutators invading mutator populations also exhibit sign inversion. It's concave down. So I didn't show you that. I showed you mutators invading non-mutators. It's concave up. Non-mutators invading mutators also exhibit sign inversion. They're favored at small populations and become disfavored at large populations. Similarly, negative time dependent and negative frequency dependent selection models also show sign inversion. It just becomes more of a menagerie of models to go into those details. But it doesn't matter whether S is positive first and negative later or vice versa.
Okay, there are no more questions on the list, but I would have one myself, I guess. Several of these relationships between fixation probability and population size look similar, right? And I mean, from your title, I would have thought perhaps, or you haven't, but there could also be a density-dependent effect on selection, like RNK selection and things like this. So I'm wondering, have you been thinking about how, what's the minimum amount of information that's needed to distinguish these things? You cannot distinguish lots of them just based on these fixation probabilities versus n, because several are concave up and look the same, basically. So you need some further information. In terms of inference, have you been thinking a bit about this?
No, I haven't. I hadn't even contemplated the possibility of a question there. Your question is, if I go to nature and observe sign inversion and it's concave up, what else do I need to do to figure out the mechanism? What other measurements do I need to make?
Something like this, yeah. Not even in nature, perhaps in your experiment, but you don't know what's causing it.
Yeah, it's a fascinating question. I also think, and this kind of comes back to some of what we talked about earlier, I would be surprised if density-dependent models, I'm thinking, won't also somehow lead us to sign inversion. I'm thinking about a simple logistic model where the model pushes counts down above carrying capacity and pushes counts up below carrying capacity. And I mean, that's not what I've said so far isn't the model of evolution. There's only one genotype, but I suspect it wouldn't take more than an hour's playing to figure out a way to build. You maybe already can see how to build a model where you build in that kind of ecological framework and again see these kinds of results.
All right, so yeah, perhaps we should stop here because we're running out of time. So thanks again for the talk and for the discussions. So just remind everyone that Dan is here until end of May and perhaps in the summer too, you said?
Well, we're hoping to go someplace else in the countryside and have more of a vacation, but you know what it's like. We tend to always be working in some sense. So, so yes, but please, you can find my email if you go to the Brown University website, or you can write an email to Guillaume, who will forward it to me, or to Louise, or any of the other folks in the organizing committee. I would love to hear from anybody. Thank you very much for the opportunity.
All right, and so next week we are having a seminar again at the regular time with Alejandro Cuse. So have a nice weekend. Bye.
Bye-bye.
Bye. Thank you very much.